My name is Dr. ABID, and today we will study a new lecture which is called Lymphoid System. So let's begin. So lymphoid system consists of B cell, T cell and natural killer cells. So here is a diagram which is we already studied in the hematopoietic lectures. So we studied that pluripotent stem cell divided into two lineage, which is common lymphoid stem cell and common myeloid stem cell. So common lymphoid stem cells are divided into natural killer cell, T cell and B cell. These are pro natural killer and pro T cell and pro B cell. They are divided into these three type of cells. So they go into bloodstream. You know that already, that T cells are mature in the thymus and they come back again in the bloodstream and settle down into different places like spleen and lymph node, etc. And we already discussed that T cells are divided into two types, T helper cells and T cytotoxic cells. We know that difference, that T helper cell have CD4 protein and T cytotoxic have a CD8 protein. That is the difference between two of them. Then T helper cells are further divided into T helper 1 cell and T helper 2 cells. So T helper 2 cell, I said that they help to mature B cell into plasma cell, and plasma cell we know that secrete antibody. So and T helper cell, what T helper cell do, that T helper cell help in maturation of which is also called macrophages, and in some places they turned into epithelial cells, and some other places they turned into joint cells, which multiple monocytes fuse together and make multiple nucleus, one big cell. They are called joint cell. So these I already discussed that. So in this lecture I will discuss these specialty three type of cells: natural killer cell, T cell and B cell. First I will discuss about T cells, then B cells, and in the last some few points I will discuss about the natural killer cells. So let's begin. But before I really go into study of the lymphoid system, I have to discuss about little bit the myeloid system player, that is neutrophil, then how neutrophil work and how macrophage will work. So how the neutrophil will work, this is a cell of a neutrophil, and we know that this neutrophil will take a star antigen. These are the star antigen. Let's say this is a star microbe that engulf by the neutrophil, and they fuse with these green bag of enzymes, and they are called lysosome, and they fuse with that lysosome and digest and make a different antigen. So these antigen are thrown into the lymph stream, and where the macrophages pick these antigen and present to the T cells with MHC2 molecules. So this is a simple diagram, and just I said that in this explain that diagram, and the same which I write here, that neutrophil are most abundant cell, then other WBC, they are most motile. When bacteria come into tissue, they come first cells they come, though they are the neutrophils, because chemotactic substance which attract the neutrophil, they release first. So what they will do, neutrophil digest those bacterias, yeah, and release its antigen in the interstitial fluid into the lymph node, where the macrophage will pick these antigen and process with MSC2 molecules and present it to the T helper cell. This is simple. So how will macrophage will work? Same process, that macrophage also digest the bacteria and make antigen and process with MSC2 molecule and present it to the T helper naive cell. This is very simple diagram. So this is a very simple diagram. Don't worried about that. We will go step by step. That this diagram show that how the macrophage present antigen to other cell like T lymphocyte. So this blue is a macrophage structure. This blue circle represent the macrophage, and we will start a story from here. A star bacteria enter into the macrophages and just fuse with lysosome. These this antigen, or we can say that there's bacteria which engulfed by the macrophages, and this circle is representing the membrane, which is eventually during phagocytosis it took by the this circle. So this will be fused with the lysosome, and lysosome, you know that they are catabolic enzyme, and they digest that bacteria and make a star antigen. And this is here is the one part of the story. The other part of story is that macrophage have a nucleus that on the chromosome number 6 have a gene. You have a special, you can say that DNA. One is shuffling DNA and other is the constant part of DNA. So every time the shuffling part shuffle, and when this part of DNA translated into protein, like I am representing here 3 protein, 1 2 3 here. So they give a peptide. So when it fold, it give a different molecules. You know that difference between these 3 molecules, 1 2 and these 3 molecules. The shape of this part, this is a Y shape part, here is a like part like this, and you can say that there is a cup shape part. So every time they give a different molecules. Every time they shuffle that DNA, it will give different molecules. So those molecules are I am representing here. Those molecules are MHC class 2 molecules, and these molecules bind with the antigen, and eventually that is represented on the surface with the antigen. So here is the class 2 molecules, that green molecules I am showing here, and here is the antigen which is processed by the macrophage. These 2 molecules are presented on the surface of macrophage. So they presented to the T cell. So that's very easy. So I was talking about this, that the antigen is processed by MHC2 molecules and presented to the surface. So these MHC2, what are these molecules? That these are a major histocompatibility molecule. MHC mean M for major, H for histocompatibility, HC for histocompatibility molecules. They are two type. One is class 1 and second is the class 2 type. We are discussing that here, the class 2 molecules. So what they are, actually they decide the compatibility of the tissue. So for example, if one person have a MHC class 1 and class 2 molecules similar with the other persons, so we say that they are compatible and tissue is resemble with each other, and they can donate one kidney or one other organ to the other person. So MSC2 molecule determine the tissue compatibility between different person. For example, if one person can give kidney to other or not, what it will decide that if the tissue is resemble or not. So how we know that the tissue is resemble or not? We see the MSC class 2 molecules. So tissue resemble if the MSC2 molecule are same in a different person. So they can give tissue to. So let's begin how the T lymphocyte. So here our lecture start that how the lymphoid system work, and what are the components. We already discussed that T cell, B cell and natural killer cell. So here we will discuss that about 1 minute. Here we will discuss about the T lymphocytes. So here are the few factor that for example, I give you example. This is a cell. This cell have a receptor here. So these receptor are called TCR. Okay. So for example, this is a cell, a T lymphocyte. These on the surface they have a receptor, they are called TCR, and these TCR will eventually bind with MSC class 2 molecule and antigen complex. So on the surface of T lymphocyte present receptor TCR, which is bind with the MSC class 2 molecules plus antigen complex. So different T cell have a different TCR. It means that, for example, this T cells have a TCR that is a cup shape, maybe other T cell which have a V shape TCR. So different cells have a different TCR, but you know that all TCR are same in one T lymphocyte. So in one T lymphocyte all TCR will be the same. So here will be, I am showing V, but for example this is one T cell, so here we also is a cup shape TCR. So different T cell pre-programmed to recognize different antigen because they have a different receptor. So another receptor which present on the surface of T helper cells, that they have a CD4 receptor. What it may mean by CD4? Actually The scientists, when they are recognizing a cluster of cell, so they see that on specific cluster of a cells have a present same protein. For example, protein number 4 uh, present on specific cluster of cells. So they give that protein name cluster designated protein number 4. So different T cells cluster have a different CD receptor, means cluster designated receptor. For example, T helper cell have a CD4 receptor, and T cytotoxics have a CD8 receptor. Just CD means C for cluster, D for designated, 4 mean protein number 4. So on the surface of T cells, which are have a specific cluster, so they have a protein on them. So that protein is called CD4, mean cluster designated protein number 4. So they are all T helper cell. So that's a simple I write here, that different cluster of T cells have a different function, and these different cluster of T cells have a different surface protein. So that's why they have a different function. For example, one cluster have one type of protein, so doctor call this cluster the protein as a cluster designated protein number 1, for example CD1. So the T lymphocyte or T helper cell have a cluster designated protein number 4. So cluster designated protein number 4, just this is the simple definition that what is CD4. CD4 are the receptor which recognized by MHC class 2 molecules. That's very important point, that MSC2 molecules, which is presented by the macrophages with antigen complex, they bind with CD4 receptor of T lymphocyte and TCR of the T lymphocyte. So don't worry, we have a diagram here. So this is our diagram. We know that this black representing black circle representing the T helper cell here. So this is a macrophage. This I am representing a macrophage here. We know that the chromosome number 6 have a specific gene which is shuffling, and every time they make a different MSC molecule, these Y shape, I am showing that they are the MSC molecules. And we know that already that antigen is processed by lysosome, and lysosome will so this bacteria fuse with the lysosome and digest uh, with lysosome, and this star antigen is representing here is the bacterial antigen, which is presented by the MHC2 molecule to the T helper cell. So what is here happening, that these antigen will bind with the TCR. We already discussed that TCR, the T cell receptor, which is bind with those antigen, and here is the CD4 receptor, which is bind with MHC class 2 molecule. So what is happening here, the CD4 receptor bind with class 2 molecule, and this star antigen will bind with the TCR receptor. so both receptor will be activated, and this will send a signal to the nucleus. So this is the one part of story here. The second is that after this binding, that from macrophage B7 protein will bind to the CD28 receptor of the T helper cell, and it also gives the signal to the nucleus. And after giving that signal, the third signal is from this, that the macrophage produce interleukin 1 molecules, and those molecules will bind with the receptor of IL-1 receptor. Here I am showing, and this will also give to the signal of nucleus machinery here. So nuclear machinery will activated, and multiple gene will be activated. What one gene will activated and give a receptor here, that gene is translated into specific messenger RNA, and messenger RNA will translate it into protein, and this protein will be here will injected on the surface of on the membrane of the T lymphocyte. So this is the receptor for IL-2 receptor, and one other gene will be activated, and this will translated into the IL-1 molecules. So after these translation, the IL-2 molecule will bind its receptor here, and it will activate another green gene here. When this gene will be activated, the proliferation of the specific colony of T helper cell will be activated, and we say that the clonal proliferation start here. So I just discuss these 5 steps in that diagram, 1 2 3 4 5. First is clonal selection. What is meant by clonal selection? That specific clone of a T cell will be selected. For example, here in the T helper cell, which one? T helper 4 cell, which have a receptor, that will be selected by the macrophage and presented the antigen. After that, the primary interaction begin. What is primary interaction? Primary interaction means that the MSC class 2 molecules and antigen will bind with the TCR and CD4 receptor, and that will give signal to the nucleus. This is called primary interaction. And then after that, the co-stimulation is start. Co-stimulation means that IL-1 is producing by the cell, and that will act on a cell which is macrophages. IL-1 produced by the macrophages, and that will stimulate the receptor on the T helper cell here. That is called co-stimulation. So co-stimulation will start, and after that T helper cell will go to the autocrine pathway. What is autocrine pathway? I just move to the previous diagram, that autocrine mean one product produced by the same cell and act on the same cell. For example, this is IL-2 produced by the T lymphocyte, and that will act on the same T lymphocyte that is called autocrine. So autocrine, this T helper cell is going to the autocrine pathway. This is called autocrine pathway. And the last one is the clonal expansion. After that, specific gene that I said, that specific gene will activated, and this will go the clonal expansion. So I mentioned here clonal selection. First pathway is clonal selection, that the helper 1 is selected by the clonal selection selected. The second pathway is that primary interaction. They interact here MSC class 1 class 2 molecule and with antigen. They interact with CD4 and TCR. This is called primary interaction. This is step number 2. Here I write step number 2 here. And after that, co-stimulation will occur. The third pathway is here, that IL-1 will act on the T lymphocyte. This IL-1 will act on T lymphocyte and it will stimulate here. After that, the third step is that autocrine pathway. The pathway is autocrine pathway, that one IL-2 produced by the same cell will act on the same cell. So this is the third pathway. After that, the autocrine pathway, the clonal selection will begin. That specific lymphocytes is selected, and clonal selection begin. This is, I am just showing one cell, but in fact lot of cells, a lot of antigen is presented by macrophage, and lot of cells will activated, and that cell is selected by the macrophage. Then that will eventually begin process of clonal expansion, which I am not showing here. The fifth process is that clonal expansion. So for example, clonal expansion means that the lot of this cell will proliferate into lot of cell. This clone of T cell will expand, and clonal expansion will occur. So this is a very simple process how the T lymphocytes are working. So next story is about the B lymphocyte. So the second part of the story is the B lymphocyte. So we will discuss that, how the B lymphocyte work. Just a few things about the B lymphocyte. That B lymphocyte mature in the bone marrow. First point is that, and every B cell is pre-programmed for producing a specific unique type of antibodies that will act on the specific antigen. Actually here the B cell which mature into plasma cells with the T helper through cell, they mature into plasma cell, and plasma cell will secrete antibodies. So they are the plasma cell actually which is secreting antibodies. So that antibody will react with the specific antigen and neutralize that antigen. So same as the TCR receptor present on the T cells, here are the BCR. BCR mean B cell receptors. So the name shown shows that B cell receptor present on the B cell, mean BCR present on the B cell, which act with the antigen. So BCR are the B cell receptor which act with antigen, and that antigen which is thrown by neutrophil or macrophage freely. So every B cell produce different type of BCR, but one B cell have a same type of BCR. Same which we discussed, that TCR, every TCR is same on the same T cell. So here the statement says that every BCR is same on the same B cell, but BCR are different in different T cell. So here are the point which is very important, that BCR, also called surface immunoglobulin, which usually IgM or IgD. So these are the antibodies actually which is present on the B cell, which are not yet secreted. So they become a receptor on the B cell, which is called BCR. They usually is IgM antibodies and IgG and IG. Here I make a comparison between BCR and TCR. We know that BCR mean B cell receptor and TCR mean T cell receptor. That's very simple, that the TCR present on the T cell, BCR present on the B cell. So that's very easy. So BCR are the B cell receptor present on the surface of B cells, and it bind with antigen which is thrown by neutrophil freely in the lymph node. So TCR are the T cell receptor which present on the surface of T cell. It bind with the antigen which is processed by MHC molecules, i.e. macrophage represent these antigen to T lymphocyte. It is easy that we already discussed these bullets in the previous. So how the B cell, i.e. plasma cell, producing antibodies. So how the plasma cell will produce antibodies actually then. This is a very simple diagram here, that the antigen is bind with these B cell, and this B cell will bind with this antigen that will activate the specific gene here, and that gene what it will do, it will do the proliferation of that B cell. Clonal proliferation of the B cell will occur here. So these are the different protein which give the signal here. For example, this is the antigen will bind with BCR. That BCR here in the inner part of the membrane have a receptor for CD79, and that CD79 will give the signal. So how the B cell, actually the plasma cell, produce antibodies. So these are the mature, I can say that these are the plasma cell which have antibodies, IgM or IgD bind with the antigen, this star antigen, and what it will happen that if this antigen bind with BCR, it will activate the protein which is inside the cell, inner surface of the cell membrane, which is called CD79. CD79 will give signal to the nucleus, and specific gene will activate it. Moreover, that uh, B cell growth factor and IL-4 from the T helper 2 cell will also bind with the receptor which is present on the B cell. So proliferation will happen. So lot of B cell will proliferate, which is specific to that antigen. So moreover, same from the T helper 2 cell, IL-5 and B cell differentiation factor will act on this proliferated cell. So B cell differentiation will happen. So B cell will be differentiated into plasma cell, and these are the plasma cell which secreted antibodies. So what antibody will do? They activate the complement system. Number one, they activate the macrophages. These macrophages have receptor for the antibodies, and one side it will bind with that receptor, and other side bind with bacteria, and they will phagocytose. So very important. And what the other function is that this antibody will activate the complement system and eventually kill the bacteria. We have a separate lecture on antibodies and separate complement system, so we don't have to worry about this. So just believe me that this antibody will help to phagocytose, and antibody will help to activate the complement system and kill the bacteria. So I just repeating that B cell proliferation will occur if IL-4 is present there. B cell growth factor, IL-4 is a B cell growth factor. So, and also IL-4 and B cell growth factor, another BCGI, BCGF also are the B cell growth factor. So these two component, they also are the B cell growth factor, IL-4 also, and BCGF. They activate the B cell to proliferate. And B cell differentiation factors, which are the B cell differentiation, how the B cell differentiation will occur, that if present IL-5 molecules, so IL-5 proteins are the B cell differentiation factor. Differentiate into plasma cell secrete antibodies. We already discussed that. And antibody, what the antibody is doing? Antibody have two main functions: help in opsonization and activate the complement system. We already discussed in the diagram before. So this is the story of the B cell. Now our last part of story is begin that B cell, the third part, which is natural killer cells. Our third part of the story here is that. So how the natural killer cell will work? So natural killer cell have a specific molecule which is called granzyme. They have a granzyme, which are the digestive enzyme, which is packed on the membrane, and these granzymes, like as a grenade which is thrown by the macrophage to the bacteria. So bacteria will kill natural killer cell will kill the bacteria. So important in the innate immunity. Especially against intracellular pathogens. So natural killer cells are larger than B and T cell with distinctive cytoplasm because they have a lytic granules called perforins and granzyme. That's very important. One is granzyme and another is perforin. Perforin are just a protein that perforate the bacteria. For example, this is a bacteria, so 5 different type of protein will join and make a hollow protein like a pipe, and this pipe will inject it into that bacterial surface. So osmotic lysis will occur. So osmotic lysis will occur and eventually bacteria die. These enzymes are digestive, and they are the digestive enzymes which digest the cell membrane, and eventually lytic will occur. So they act on a target cell process. Just the same thing. Actually the apoptosis means that a programmed cell death will occur. So distinguish between healthy and infected cell. These natural killer cell will distinguish between the healthy and infected cell by recognizing the cell surface protein induced by stress, malignant transformation, or microbial infection. So induce apoptosis in the cell if they don't express MHC class 1 molecules. This slide is just saying that if the cell, bacterial cell or our own cell or any malignant cell, they don't express MSC class 1 molecule on this surface, so a natural killer cell recognize that is a foreign cell and dangerous cell. So a natural killer cell will kill, and a programmed cell death will occur, which is called apoptosis by the secreting of perforin and granzymes.